Welcome, everyone, to our hybrid meeting for our Frontiers in Biostatistics seminar series. For those here in the audience, we will be answering questions. You will be able to ask your questions yourself, and those who are on Zoom, please post your questions to the Q&A tab, and we will be keeping an eye on them and then read them and hopefully get answers. So today, our speaker is Dr. Bill Lauder. He's an assistant professor at the Department of Data Science at Dana-Farber, as well as at the Department of Pathology at Harvard Medical School. He has his bachelor's degree in math and as well as an integrated sciences. He also has a master's degree in applied math from Northwestern University, which he earned in 2010. He also holds a PhD degree in biophysics from Harvard University, which he earned in 2017. Dr. Lauder's expertise is in artificial intelligence and machine learning. He's specifically interested in biomedical application, but his expertise is not limited to that. He also worked on the algorithmic trading and sport analytics. He was a co-founder and a chief technical officer for the Deep Health. It's a company that successfully developed and deployed two FDA-cleared methods for early breast cancer detection. Those methods are used for early breast cancer detection using mammographic images. In 2022, Bill decided to return to academia and joined our department. And here he continues to work on the artificial intelligence and machine learning. He's developing robust algorithms of computer vision, taking lessons from the human vision, and he's interested in further implementation of those methods in the clinical science. Today, he will tell us about the current state of artificial intelligence in medicine and the future opportunities. Thank you very much.
Awesome. Thanks, Svetlana. Super great intro. Better than I can do for sure. So yeah, as Svetlana mentioned, so I've been here about a year now in Department of Data Science. And before that, I'd actually done a startup focusing on mammography. And so today I'll be talking about a little bit of work along some of these directions and trying to give a general sense of the current state of the field as I see it in AI for medical imaging, as well as some future directions our lab and others are working on. And so to start, if I can get my mouse to work, nope. Oh, there we go. Okay, so just kind of level set when I'm talking about AI, specifically talking about kind of deep learning, and as just a kind of distill what we mean by deep learning. Ultimately, a deep neural network takes in some type of input and it's going to make some type of prediction. And so whether it's a kind of a foundational model or a convolutional neural network, I'm going to be talking more about kind of the outputs today and what we deal with it, and less about the internal structure. And the kind of crux of deep learning as it's evolved over the years is this notion of end-to-end optimization. So in the context of, say, mammography, which I've worked on, maybe we want to develop an algorithm to detect breast cancer. And the real crux of deep learning is getting a lot of data and training this network end-to-end. So essentially updating all the layers in the network to perform this task. And the reason I kind of start with this simple illustration is I like to use this metaphor of end-to-end optimization as kind of a cheesy analogy of how I like to think about applications in general. So if we zoom out, the algorithm development for any application is just one step in the full pipeline to clinical integration. And ultimately, if we want to have an effective kind of clinical product and make it to patients, we really need to think about this end goal of clinical integration from the start. And one thing I particularly want to hit home today is this notion of intended use. So the intended use, what it essentially means is, how do we actually want to use this product in the clinic? What type of data is it going to process? What is its outputs going to be? And ultimately, how is a clinician going to use this product in a clinical setting? And so one thing my group's been interested in recently is you hear more and more about maybe FDA clearances and different AI algorithms and AI in clinical practice. And we've been interested in kind of summarizing where we are in that state of the field, and particularly around this notion of intended use. How are things actually being used today? And so in a project with Stephanie McNamara, who's an MD PhD student here, we recently went through essentially all the FDA clearances for AI products for medical imaging to try to summarize how these are being used. And so long story short, there's a bunch of different databases that the FDA has. We've gone through these different databases from PMAs, de novo clearances, and 510 PMA pre-market notifications. And essentially for each product, if you go on any of these databases, you can get what's called a summary. Essentially, the public information of what is the product indicated for, what is the data that processes, these type of things. And so we've gone through all these, a couple hundred at this point, and tried to summarize what, how things are being used. And through this, we've seen that there's essentially 5 different broad categories of FDA cleared products for medical imaging in the context of this term CAD. So CAD is a term that's kind of evolved over time, but it initially came from this nomenclature of computer-assisted detection. And now across the list of FDA cleared products, there's essentially 5 different CAD types we see. So the first type, which is actually the most popular clearance at the moment, is this CAD type called CAD-T. And what CAD-T is essentially a triage device, where the idea is we're simply going to flag exams for prioritized review. So in the context of mammography, you can imagine taking an AI algorithm, and it's going to score exams that are particularly suspicious. So maybe a radiologist can review these exams sooner. The upshot here are these CAD-T devices essentially output a binary indicator. Is the exam suspicious or not? There's really minimal kind of localization and outputs. It's just this binary indicator. Next product type of product that's been around for a little while now is this notion of CAD-E, which is meaning the E stands for detection. So these products are designed to help aid in the detection of different lesions. So in the case of mammography, it might put a bounding box over a particular lesion. And so now beyond CAD-T, it actually provides some localization and not just a study level score. CAD-X, on the other hand, sorry for all the word soup, but CAD-X is designed to aid in the diagnosis. And so here, it might help a radiologist to actually make a determination, is breast cancer likely to be present? Should we recall this patient for further testing? And the notion here is it provides maybe minimal localization, but it gives some more fine-grained output, say a score between 0 to 100 of how suspicious the exam might be. And then there's also a version that's CAD-E and X. So it also has this high-level score, as well as, say, localization around suspicious lesions. And then finally, for one particular indication, which I'll go to, I'll talk about it a little bit, there actually are products that essentially provide an automatic or kind of autonomous diagnosis. Again, it's really limited to a particular application, but here the idea is not just to kind of aid a clinician in making the decision, but maybe making the first kind of initial decision itself to then forward the patient to further follow-up. So these may seem kind of arbitrary and what are the lines between them? But essentially across all the different disease types that are currently out there, each product that is currently FDA cleared falls into one of these 5 different categories. And so, like I said, we've gone through all these summary statements and try to see where things fall. And what we see is that actually the majority of products that are cleared are these so-called CAD-T devices. So again, these devices only output essentially a binary indicator to help triage a patient. And overall, there's been 100 unique products of the CAD variety since 2016. And after CAD-T, you see some representation of each product. So CAD-E, the detection, CAD-EX, CAD-X, and this autonomous version as well. And so these are kind of the CAD types. if we look at the diseases, which you can't see from this thing, let me scoot this over. Okay. There's definitely a number of kind of disease or conditions that are indicated for with products at the moment. So here's just a distribution. I don't expect you to look through all this at the moment, but color-coded by the CAD type. And what you'll see is, the most popular applications are intracranial hemorrhage, such as like a stroke detection. Breast cancer actually has the same number of products of 14 and has a representation of different CAD types. But overall, a lot of the most popular products are populated by these triage devices. So things like stroke, pneumothorax, where you can imagine it might make sense to have these triage devices. These are, urgent cases you might detect in, say, emergency department, and you want to flag these for a prioritized review. And then, as I mentioned, there is one indication where there is somewhat of an automatic, autonomous diagnosis at this point, and it's specifically for diabetic retinopathy. And this, essentially how it works is you have a fundus camera, and the algorithm will automatically detect whether the patient should be forwarded to a specialist to kind of work up the diabetic retinopathy. So that's kind of a landscape of where we're at. And as I mentioned, the majority of products so far have been CAD-T, this triage. And that brings back to this important question of intended use. How are we actually intending to use these products in practice? And interestingly, there's been some kind of questions or at least feedback from the FDA recently that we really need to focus on these aspects as well, really hitting it home. So for instance, a letter earlier this year, the FDA published this letter to healthcare providers to really call to attention, hey, these triage devices, you need to focus on what they're indicated for, which is flagging exams for suspicious findings. They're not intended to kind of replace a full interpretation. Importantly, they don't flag out negative exams. So they're essentially, you want to prioritize, this exam looks suspicious, let's review that. But you can't ignore the so-called negative exams. And importantly, if it's used as intended, it can help improve workflow, but particularly calling out that these are not diagnostic devices. And so while these kind of, different letters, T, X, E, A, may seem like word soup, it's becoming clear that we really need to focus on these things as they're integrated. And how this also relates to an important concept of what type of testing do you need to get these products cleared. And so if you look at triage devices, these CAD-T, the core pivotal test you need is actually standalone testing. So you're not testing, say, how does the clinician interact with this product, because it's really designed to just prioritize exams. This is in contrast to these CAD-E, CAD-X, and CAD-EX devices, where they're really a to assist the clinician as they're actually interpreting the exam. And then finally, on the autonomous side, currently the current indication, you need a prospective standalone study. So as we think about integration and trying to simulate how do clinicians actually use these things, that's where these reader studies come in. And so I'll talk about today a reader study we've done at Deep Health, looking at how we can actually optimize a specific application, and that's screening mammography. Go ahead, James.
How do laboratory design tests fit into this? And like maybe, I don't know if there's an equivalent bar chart you have somewhere, if that's even possible.
Yeah, it's a great question, and I should be clear about kind of the boundaries of this analysis. So what we looked at so far is things that are like have a CAD indication, so something that assists or aids. There's other avenues where you can imagine trying to quantify a known quantity, so maybe you're measuring the size of a lesion, but not kind of assisting the interpretation, or in the context of maybe pathology of having a ingrown kind of laboratory test you're doing. I think there's a murky line there, and it's unclear, at least to me at least, kind of where they fall in the FDA purview. I think historically, one line in the sand is if you have a algorithm that is, say, crossing state lines, where you're doing it's kind of this interstate commerce kind of flags you fall under the FDA's purview versus kind of just in-house test. And so I think it's an evolving area. But yeah, this is at least in terms of the FDA clearances, and they're so I'll get to it later as well. But the vast majority of these products are on radiology-based imaging, but there is one product currently on pathology imaging as well. Okay, and so to just kind of further beat a dead horse and bring home of kind of thinking about this end goal, what is the intended use? So when we were thinking about mammography, part of it comes down to detailed understanding of what the workflow is like. In the case of mammography, a radiologist would interpret a screening mammogram, and ultimately the core decision they need to make at this point in time is, should I recall the patient or not for additional testing? So it's this is the core decision the radiologist would need to make. And again, so when we set out to design our product and algorithm, it's ultimately to help with this really binary task. There's 100 different things we could try to do, but let's really focus on this end goal. And ultimately, to borrow on more kind of FDA terminology, is kind of what are the software requirements, or what are the requirements we need to do from this? So ultimately, we want something that's actionable, that can help a radiologist make this decision. We want it to fit seamlessly into a radiologist's workflow. We want to do other things of not only output a study level score, but also localize lesions that ultimately form this recall decision, and so on and so forth. So I'm going to kind of skip over some of the nuts and bolts to get to more of the later things, but if you're curious, so we have developed an algorithm that goes under the hood and serves as a basis for this product. I'm happy to talk in more detail if we want. But I want to focus on is, the algorithm development is these first 3 steps. There's so much more beyond that. And so things that may seem somewhat mundane or kind of details that you might not necessarily care about, actually come to play a big role when you're actually trying to clinically integrate. So ultimately, we took our algorithm and developed a product called SageDX. And this product is designed to be minimal. When a radiologist is reviewing a mammogram, they can simply click a button, the outputs of the product come up. And these outputs consist of bounding boxes over lesions, as well as what we call a suspicion level. And so, many algorithms these days, and especially in research contexts, we have some algorithm that outputs a score between 0 and 1. When talking with radiologists and thinking about this and workflow, it becomes clear kind of what do you do with a score of 80 versus a 79 versus a 60. And so what we did is try to develop categorical outputs that are designed to aid in this end goal of recall versus no recall. So at the highest level suspicion, which contains maybe roughly 5% of cases, we call this high suspicion, where the algorithm would essentially recommend recall for these patients. At the other end of the spectrum is kind of a minimal suspicion, where in our testing, we've seen really low number of cancers. So the algorithm is essentially fairly confident that these patients aren't deemed necessary for recall. And then in between, we have an intermediate and low categories, where in the intermediate cases, this is where maybe half of these cases might be suggested for recall based off the radiologist's decisions. So things like this, aligning with our requirements, kind of having these task-aligned categories that we can click on and off, provide localization as well as case and lesion level outputs. And then coming back to give a sense of what reader studies look like. Again, the goal here is to test how clinicians actually use the products, in addition to just testing the standalone performance of the device alone. And how reader studies work is typically you have a set of radiologists or clinicians who read the exams without the AI's help. And then again, with the AI's help, it's kind of a mixed design. And ultimately, you want to compare the performance between the two. In our case, we collected 240 cases from sites we had never used for development, with 18 radiologists who are unaffiliated. I'm going to give a high-level summary of the results. Happy to go in more detail. But ultimately, what we saw is that when radiologists actually use this device as intended, with our kind of actionable categories, we saw that they, on average, improve both sensitivity and specificity, with a reasonably large jump in AUC as a kind of summary metric of an ROC curve. And we saw that, in fact, all 18 radiologists improved when they used the AI in this setting, where the standalone performance of the AI alone was actually slightly higher than all the radiologists without the AI. But when the radiologists actually used the AI, many of them performed higher than the AI alone, kind of putting this potential of AI plus human is better than either alone.
Question. So when you measure or calculate the AUC, do you do this? This was not in a clinical trial?
So it's a retrospective setting. So they output essentially a binary decision as well as a kind of 0 to 100 score. And so there's a big caveat for reader studies is it's still not clinical practice. So it's kind of one step towards getting to clinical practice. And so that's where, to me, there's no perfect metric for any of these type of things. But for instance, sensitivity and specificity is measured based off their binary indication of whether they recall a patient. And then kind of the ROC curve is based off this continuous score.
And so my question was, how many patients have you used, or how many individuals were included? And was this based on a sample size calculation? Or
yes, sorry, I kind of skipped over some of those details, but in this study, there's 240 cases. We did a power analysis where that would be sufficient with 18 readers.
How did the compare of AI versus AI plus human reading?
Yeah, great question. We saw that the AI alone actually performed better in this setting than all the radiologists without the AI. But when we combined them both, there was a number of radiologists who performed better than the AI alone when combined with the AI. Yeah.
What are cases that you started going back to that, the cases that consecutive cases, or did you enrich on the findings?
So it's a good question. It's consecutive, but with oversampling of the positive cases. So one challenge here, and again, I'm kind of skipping over the details, but the incidence on a screening exam of breast cancer might be 5 out of 1,000. So if we really wanted to test that prevalence, we need a super long reader study, which isn't feasible. So typically what you do is you have some period of time and you collect things kind of sequentially for the positives cases. Oh, and then for the negative cases, you kind of randomly sample over that period to have reasonable distribution. But it's a great point that even this reader study is a simulation of the intended use. So it's one step beyond kind of the standalone testing, but it's not the full evaluation. Another aspect on reader studies. So people have seen that, certainly with enrichment, their radiologists perform differently. But we've seen that, or at least in other studies, that oftentimes this can represent more of a shift along the ROC curve rather than a new ROC curve altogether. But definitely a caveat. And so just kind of bringing things home we ultimately got FDA clearance for this product. It's one of these 13 CAD-EX products. And in terms of actually measuring things in clinical practice, so we have since, we ultimately got acquired by this company called RadNet, who's a big kind of imaging services provider, where we've deployed our products and they're processing about 1 million mammograms a year at this point. And fortunately, so it's no real fully published data yet, so you have to take it with a grain of salt. We do see that the results we've seen in the reader study do tend to hold up in practice as well as we've monitored. But more to come there. Okay, so quick pause. I wanted to really, go ahead.
Is there any prospective clinical trial being run on it?
Yeah, it's kind of a good question. We yes and no. We don't have a fully randomized trial that we've done, but we've measured kind of pre and post deployment of the AI. So not exactly a randomized clinical trial per se, but there are other RCTs for other products in Europe mainly, where there's kind of a double reader paradigm, and those are starting to show promise as well. So I will say, as we kind of loop back things into the interim summary, I really want to emphasize this point that of thinking about the intentional alignment with the intended use. And where we currently are as a field is that the current FDA cleared CAD use cases are essentially triage, detection, and diagnosis with some variations, where most of these products are CAD-T that have these binary outputs. And there has been some pushback recently of like, we really need to make sure these things are used and intended and think about how the clinical integration comes into play.
So for the diagnosis accuracy and performance there, have you noticed any performance improvement in the clinical side in terms of resources and how much time the radiologists are spending?
Yeah. That's another great question. And one, I'll say we have seen some evidence, but it's really hard to measure. You would think it should be relatively easy to measure. It's how long do you spend on scans, these type of things. With all this, and I think one kind of take home message of where we are in AI, people have pushed on the kind of productivity. That's a reason to use AI type of thing. When you look at, say, a radiologist's schedule, and I want to preface, I'm not a clinician, but I've kind of seen different schedules for people that, they might spend an hour here reading screening exams, and then they're doing a biopsy, then they're doing blah, 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 blah. And if you save, say, 25% reading some of the exams, it's easy to fill that with like another call, or like they need to do another biopsy, or something else comes in. So there's two things, like how do you explicitly measure, like when do you start and stop the exams? There's a lot of infrastructure for that. And then two, how do you measure that in terms of overall productivity when other things kind of creep in? So I think that's where it's become less clear. You really get those big gains focusing on, say, individual applications. And maybe that's where things like the autonomous detection, where it's like clear, like, a radiologist doesn't look at it, I think could maybe move that along, but there's a lot of considerations there as well.
Also, you have to compare behavior that is optimized for one setting and that would have to be adapted to another setting. So if it isn't adapted, then you probably won't gain
That's absolutely the other thing. One thing we found almost paradoxically is that some of these AI solutions can almost make you rethink how you do your workflows generally. So maybe what you need to do is less, people call it kind of like batch reading. You sit down and you focus on like one thing and knock it out versus kind of interleaved. And that's almost an AI independent thing. But the AI can move the needle to force you to want to do that. And for instance, where if you can rank exams in terms of suspicion, you can like maybe think that batch reading might help differently if maybe you want to start with easy exams in the beginning of the day, harder later in the day. And so I think, as we think about how we actually make these things work, it does call to attention, not just the AI itself, but like, can we rethink these workflows generally? Okay. And so I know those are kind of like, really detailed minutia, but I want to emphasize that they end up becoming important when we think through. But as we zoom out, I want to talk about some kind of like future and ongoing applications we're working on. And as I mentioned briefly, like if we think about broad scale and particularly maybe focusing on oncology here at Dana-Farber, very high stroke of what the continuum might look like. We might have screening and detection. We catch the cancer and then we diagnose this, maybe subtype the cancer. And then ultimately the goal is, if you can detect the cancer, we want to get better at treatment. And so we might be able to, predict prognosis and optimize treatment and these type of things. And so there's a lot of applications of potential AI along these workflows. For all these, there can be different types of data we use, radiology images, pathology, even EHR, text genomics, different omics. And across all these, of course, there's common challenges as well. There's big questions of bias and fairness. How do we make sure these things work for all patients? questions of explainability. How do we kind of understand what these these models are learning? How do we develop kind of clinically actionable explainability? And as we talk about, how do we kind of integrate these algorithms with clinicians? And so, again, is kind of a note of where we currently stand. If you look at current FDA clearances, all but essentially, at least in the CAD space, are mirror in the radiology imaging, or at least no in vivo biopsy images, besides one application in pathology. And these applications are also in the kind of detection and maybe diagnostic tasks. And looking at the future, there's a lot of ongoing work of kind of, can we move further down the pipeline? The thought being, if we can detect cancers better, that only helps if we can treat cancers as well. And so there's a lot of opportunities there, and especially on the kind of multimodal paradigm of, let's look at pathology imaging combining with omics and these type of things. And of course, addressing some of these challenges. And so I'd say our group is working on almost all these applications. I'm going to particularly talk about two today to focus on. The first is in the area of explainability.
I have a question in the meantime.
Oh, yeah, go for it.
Related to the previous section. So there has been some debate in the community for generalizability to different hospital and health systems for deployment of medical AI systems, including radiology AI systems. There is recent work that says we should focus on local development and validation than global generalizability. What are your thoughts on that debate?
Yeah, great question. A debate I'm not going to solve today, but I'll give you my 2 cents. So first, it's true that ultimately what we care about is local generalization. And at any given point in time, we're going to be playing it somewhere. We want to make sure it works there. I think a big question in the field is, when generalization fails, why? Can we come up with a more mechanistic understanding of why does it actually fail? It's easy to say, oh, it's a new clinical site, oh, it's a new patient population, but I still feel like we're kind of lacking a mechanistic, how is this reflected in the images? And so we're working on a couple of these things. I will say that the question of generalization partly depends on the imaging application itself. So some applications are more standardized than others. And I think mammography is one on the kind of more standardized front. For instance, we've done a number of validation tests, and for instance, our reader study, we actually took data from sites we'd never touched before, even for previous testing validation, and we saw that the performance generalized. And I think that's partly because of standardization of mammography. There's maybe a few manufacturers, there's a lot of attention to kind of quality control and these type of things. On other applications, particularly maybe in the pathology domain, where there's a lot of variation on the H&E staining, these type of things, I think there's, maybe perhaps bigger challenges by default. And so, yeah, I don't think I have a particular stance of should we focus on global versus local, beyond the sense that before we deploy locally, we need to know it works locally. And part of this explainability work I'll be pointing out comes to that as well. And so, in the context of explainability, that's been a hot topic in deep learning forever. How do we open the black box and know what a deep learning algorithm is actually using? And I would say the vast majority of explainability approaches so far have been almost answering the question of where. So in the context of mammography, we might put a bounding box over a lesion, or, if we're recognizing a dog and a cat, we might have a heat map over the dog versus the cat, and these type of things, kind of answering, yeah, this is where the algorithm is looking. But that's not necessarily how humans do explainability. If I were to explain to you why this particular lesion looks malignant, I might say, oh, it's kind of has these features that's spiky versus it's not really round, or, this is a dog because its ears are pointy. I'd use some type of language to help explain this. And then at an extreme, I can give you two completely novel classes of objects that you've never seen before. And even through a couple examples, you can tell me, yeah, whatever these greeble things are, well, they seem to be like purple, maybe have these pointy little horn things, and these grables are more yellow and kind of cylindrical. And so, you've never seen these things before. You can automatically, even if I told you the differences between them, you can tell me how they look differently. And so we've been interested in, can we also kind of hijack this strategy? Can we come up with text-based explanations for a visual task without necessarily having these text explanations to begin with? And if we can do this, can we then interrogate what the algorithm has learned? Maybe it's learned features that humans don't use, or spurious correlations, or maybe it's kind of learning things that align with human knowledge. And so how do we do this? We've been working on an approach that essentially leverages joint embeddings between text and images. And so there's this really popular model now called CLIP from OpenAI. And what CLIP does is it essentially takes an image and text and tries to map, if they come from the same example, tries to map them in the same representational space, or kind of the same vector in that space. And if you give me an image and text from another image, like a caption. So in this case, if we have a picture of a dog, and you give me a caption that says this is a cat, we want that text embedding to live in a different kind of area in the space than this image embedding of the dog. And so this is this notion of contrastive learning, kind of learning similar text and image representations. And so we've been working on this in a medical context, and with Shobit Agarwal, who's a student in the lab, actually high school student this summer, who's done a lot of great work. And our idea is, can we use this kind of CLIP model that has been trained on natural images and text in the same way that a human could then learn a new visual task and use language that they've learned to explain this task? Can we do the same thing for medical images? And so our approach essentially goes like this. We have a new task that we're trying to learn. So maybe we're trying to classify masses from a mammogram. We have this image encoder that's been learned from CLIP. And if we pass these through the CLIP encoder, we'll live in some n-dimensional space where we can learn a linear classifier to essentially classify maybe benign versus malignant lesions. And it's not perfect accuracy like anything else, but it can do reasonably well in this task. And since we live in this joint space with images and text, what we do is then take a set of words where we specifically asked ChatGPT. We said, if you're to explain general visual properties of an object, what would they be? Size, shape, maybe contrast. And okay, give me words that explain these different axes. And okay, large, small, spiky, round. And if we pass all these words through the language encoder, they're also going to live in this space. And what we've been trying to do is, can we essentially represent this linear classifier as a linear combination of words? So each of these words is going to, so let's see, you can see my pointer, word 1 might be here, word 5 might be here. Here's our linear classifier betas from the image task. Can we come up with a linear combination of the words to essentially estimate that classifier? And from that, we essentially get a weight for each word that signifies, how for more or less its correlation with the visual task. And so this is kind of a simple approach, but we wanted to see, how does it work? So we first started with mammograms, so using this public data set called DDSM. And if we look at the weights that come out of this task, we actually see, again, I'm not a radiologist, but they kind of align with clinical knowledge. So again, it's an AI talk, so I have to talk about ChatGPT. So if we just simply ask ChatGPT, what distinguishes a benign versus malignant lesion on a mammogram, it'll say things, and you'll find these things in textbooks as well, an irregular shape. So if the lesion is asymmetrical rather than round, it's more likely to be malignant. And we see that, yeah, if we plug in the word asymmetric, it kind of aligns with the malignant prediction, malignant weight. On the other end, symmetric and round is benign. Sorry, I should illustrate this better. The blue is benign, red is malignant. Other things would go down, kind of round, smooth versus pointed edges, kind of, yeah, they're generally in the right direction. Size as well, larger lesions tend to be cancerous. So, you know, at least nominally the words that come out somewhat align with clinical intuition. Yeah. Yeah. Yeah.
The opposite. I guess you would say, well, it's the thing in between. It's neither dark nor light, which is to say, but it sort of raises the issue of you know, like all of this is except for maybe asymmetric. What's the word?
The collinearity, essentially.
Varying.
Yeah.
It depends on your standardization.
Yeah, absolutely. And so we noticed this as well, and I haven't quite figured it out yet, but you could also think about, and again, this is a hypothetical scenario. You could imagine a scenario where if you're really light, it's more likely to be malignant, or if you're really dark, it's more likely to be malignant. And if you're in the middle, it's more likely to be benign. Not saying that that's the case, but that's one potential hypothesis. The other hypothesis is these are highly collinear and they just kind of cancel each other out, and it's almost a spurious correlation. So that's definitely a limitation to this initial method. And so we're working on maybe ways to regularize this and so on and so forth. But definitely a great point.
Have you tried like a sort of, you know, pairs of words? Because, for example, lights and series, that is something which is
Yeah, and we actually have done that as well. Essentially, take each pair of words and you can maybe even subtract them, or just fit them. And you do get predictive performance. And what we see is that, you know, if you add all the words, you get slightly more and more predictive performance. So it's a good question of like, what if the goal is to come up with like a minimal set of words, we might have a different approach. And so I should preface, this is kind of recent work. It is submitted as a kind of an initial paper, but a lot more work to be done for sure. And the other thing is you can also look at examples of what is it actually looking at here. And generally, you know, we can essentially compute a score of how well does the example align with that word and so on and so forth. And we see, yeah, it kind of makes sense, but sometimes it doesn't. Like if we look at round, the examples that come out to be looking highly round are, okay, this is kind of a round lesion. But interestingly here, this is this little thing is probably what's queuing on as round. To my sense, this probably looks like what's called like a nipple marker. Sometimes they put a little mark. And so here it's like, you know, you're not looking at the lesion type thing. So it kind of points out of, okay, are there maybe potential like spurious correlations in the data set we could potentially find here? Large, we can see, yeah, okay, this looks large, but there's also, you know, kind of unclear. And so what we kind of circled around is we think this approach maybe one, it can give maybe general intuition of adjectives that describe a task, but maybe also potentially be useful to identify spurious correlations, which I'll get to in a second. We also done this for a skin lesion task. And here, again, if we look at clinical guidelines of there's a thing called the ABCD rule, where, for instance, a border, if the lesion has kind of irregular, ragged borders, coarse borders, it's more likely to be malignant versus round and smooth. But at the same time, we also run in this issue of, okay, asymmetric should theoretically be on the malignant side. Is that a failure of the method, or is it the failure of the model? It's kind of a chicken and egg thing that we're exploring. And so, you know, everything I just talked about is kind of like hand wavy, right? I'm like, yeah, it kind of lines up. And if we look at explainability literature, it can be hard. Like, how do you actually empirically evaluate explainability? You might see a heat map. Yeah, that looks pretty, but how do we know this actually is explainable? So one thing we did is actually a pilot rater study. And so our thought was this: if these words are actually explainable and interpretable, well, then we should be able to give them to a non-expert, somebody who has no experience with it. And just knowing the words, say round and smooth, they should be able to maybe perform this task above chance. So we did this. We got, I think it was maybe 14 volunteers, where we first had them do the task without any of the words. So just kind of do your best. And so this is particularly for the malignant, the melanoma task I'm showing. And so if we look in the first session where they, have no experience, we give them no more instructions, but besides just classify this as benign or malignant. They're roughly at chance level of accuracy of 50%. This was a balanced test set. And in the second session, if we say, okay, here's some words that are generally associated with malignant as deemed by an AI model, and we list the top 3 words of, coarse and large, and benign is more round and smooth. We see that again, super high variance, but they do improve somewhat. So they significantly improved by about 10% accuracy, not uniformly at all. And so, we don't see this as an end all be all, but kind of a proof of concept of kind of explicitly testing explainability. And here in the context of explaining a task in a minimal setting, only just giving words of that distinguish benign or malignant. And then finally, on the kind of shortcut connection side of things. So one of the things that ties back to generalization is this idea that sometimes in a data set, you might have a spurious correlation that corresponds, that has essentially a correlation with malignancy or benign. So if we start with the case of melanoma, if we see this ruler, again, I'm not a clinician, or these markers, sometimes, a dermatologist might mark up a suspicious lesion. They might use a different kind of dermoscopic microscope to measure it. And what we see is that if we use our method and plug in the word marker and identify similar examples to this, it kind of flags things that do have like a purplish marker, or maybe the marker from a ruler. And we find 2 things. One, the word marker has a positive weight for malignant, meaning that it's correlated with a malignant prediction from the model. And indeed, if we look at the proportion of cases that are malignant that have a high marker score, it's higher than kind of the default chance, suggesting that, as you might expect, if you have these marker images in the data set, it's more likely to be malignant. And the model has essentially learned this as well and is using it. And the same case for the mammography task. So what you see here is they're like biopsy clips, which sometimes after a biopsy or before a biopsy, the location of the lesion might be demarcated. And these things we also saw had a positive correlation with malignancy, and again, had a positive correlation with the model's kind of prediction. So again, we see this method potentially helping to identify these things more efficiently. Let's see, I see a bunch of other questions. Is it likely that either the symmetric or asymmetric category is capturing or something else? Yeah, great question. And maybe if I go back to the examples, it's partly an empirical thing. Like let's look at these and see what they look at. So the asymmetric versus symmetric, to me, sometimes I could see why asymmetric would come out. It doesn't necessarily have to be looking at the lesion, but here maybe there's the shape is somewhat asymmetrical. Here potentially it's cueing on this little marker. Again, the lesion is somewhat spiky and asymmetrical. On the symmetric side of things, what's interesting is, to me, it's not as clear as is actually using the word symmetric as we would for the lesion. You could hear these things are kind of microcalcifications, and they do somewhat have a symmetric pattern, but it's partly unclear. And so this is where we definitely need clinicians to help review these things. And for instance, one area for the skin lesion is this context. The high scoring word for malignant was low contrast. We actually had no idea what this really meant, and so we had a dermatologist look at this as well. And their take was it might represent low contrast between the border and the kind of rest of the skin, which is a suspicious characteristic if it doesn't have kind of a well-defined border. But again, that's kind of, might be cherry picking, and that's where I think these kind of empirical metrics are useful. Okay, so let's see. Have a couple minutes. I'm going to do what you're not supposed to do in a talk and try to cram something in really quick. Just kind of more as a primer than any real results. But I wanted to touch on, if we go back to where we currently are and where we're headed, there's a lot of applications in kind of the treatment side of things and multimodal approaches. And so we've been working on this as well. And one project I wanted to call out, again, is as a primer, and there'll be much better talks with much more details soon. But this project here at the Dana-Farber called Immunoprofile. So this is a huge project that's been in the works far beyond since I've gotten here. So led by Scott Brodig and Ethan and many other people in the crowd have been working on this. And I think it's a really cool heterogeneous data set and what Immunoprofile is essentially a multiplex immunofluorescence assay that allows us to kind of generate a more detailed phenotype map of a tumor. So there's different markers. I don't think I have enough time to explain kind of like a multiplex immunofluorescence, but if you're not familiar, it essentially allows us to determine different cell types within a tumor, and then we can kind of learn correlative features across these things. And so, again, very high level overview of things we're looking at. One question people are especially interested in and has been shown to correlate with overall patient prognosis is essentially, how inflamed is the tumor? Do you have immune cells infiltrated the tumor? And typically, these metrics are measured based off of cell counts. So, how many CD8 positive cells do you have? CD8 positive immune cells. What we've also been trying to do is, can we quantify the spatial arrangement of these cells and see if they offer any other prognostic value? And again, skipping over details, we've come up with this metric that essentially quantifies the proximity of immune cells to the tumor. Where on the left, you can see these tumor cells in red and these CD8 positive immune cells, where the immune cells are generally really close to the tumor, kind of infiltrated the tumor. Versus the one on the right, there's essentially the same overall number of these CD8 positive immune cells, but there's a very different spatial arrangement. They're kind of excluded. And so based off of this and these density metrics, we've been trying to first describe, what are the different types of variation across tumors in this data set that's very heterogeneous. So it has a lot of different cancer types, a lot of treatment types, a lot of stages. Can we just get a better sense of describing what these tumors look like? Again, bear with me on these kind of the slides. There's much prettier figures being made, but one thing we found is that kind of clinical intuition almost comes out naturally as we look at these sources of variation across tumors. And so, for instance, we take these spatial metrics and these density metrics of the different immune cells and performed a principal component analysis to just see, what are the axes of variation. We see things of, hot versus cold tumors. The second principal component that comes out is more of the spatial arrangement. Is the tumor infiltrated versus excluded? We see other interesting things of, not only just is the overall tumor inflamed, but what is the composition of that tumor? Is it more kind of cytotoxic versus regulatory inflammation? How do the cells, different immune cells, spatially arrange close to the tumor? Again, you'll probably not remember any of this, but just kind of a primer. And based off these different phenotypes that we've identified, we've also been starting to look at how do they correlate with overall survival and patient prognosis. And we do see that not only they kind of describe how different tumors are arranged, they also can kind of correlate with this overall survival. And so one simple analysis we can do is for each of these principal components, like, are you on the good or bad side on that principal component for the particular tumor case? And if we essentially do this for all the principal components and add up the combinatorially combination, we do see that you kind of gradual more separation in these Kaplan-Meier curves. Okay, again, I can just kind of barrel through that. Much more coherent presentations coming soon. But with that, I know we're coming up on time, so I want to acknowledge all this has been a huge team effort. A lot of people in my lab across Dana-Farber, previous company, Deep Health and RadNet. And yeah, thank you for all coming today. Question?
Yeah, a couple questions regarding the presentation on foot.
Yeah.
I was wondering if you had done any work with sort of dimensionality reduction in sort of analyzing the sort of topology that latent space of this, and that you'll find in some sort of neural net is that the embedding space, like a certain dimension, might begin to correlate to some kind of semantically meaningful aspect of stimuli. So you might have like the vector actor and actress might differ exactly, completely parallel except in one dimension that might like sort of gender in a dimension of embedded words. So I was wondering like if there's been kind of dimensions that you found kind of using sort of PCA or some other unsupervised method that indicate some sort of large variance. So maybe like it would correspond. So maybe you have like 4 components of these embedded vectors that correspond to asymmetry versus symmetry or whatever. And then also if you compared maybe that to sort of the activations in the network itself, and sort of you found sort of isomorphism between those two manifolds that you learned, indicating that the network is visually sort of thinking the way it's sort of linguistically.
Yes, it's a great question.
Request to repeat.
Yeah, I completely forgot to do that. Yeah, I completely dropped the ball there. So the question was kind of understanding the topology of the clip space, and so in the imaging kind of classifier direction, can we understand sources of variation? Maybe do they align with particular words and so on and so forth? Does that capture the question in a sense?
Yeah. Like the variance in this sort of in this latent space corresponds to variance within kind of like the typical properties that a document would look for in a patient. So variation in symmetry, symmetry, variation in latent art as opposed to variation in like I don't know, like isn't that what I don't know.
Yeah, it's a good question, and I guess it's part of the motivation of the approach to begin with is kind of like, can we come up with a combination of words that align with that classification decision? It's one thing we want to try to do is do it more in a I guess end-to-end optimization sense. So here we're kind of a priori choosing words, and we particularly choose words that are kind of can describe object in general, but you can also imagine like almost generating text, feeding that in, and having some loss function of the text embedder. How well does it align with the image classifier? And then you have some loss, and maybe kind of optimizing that end to end. It's a little trickier than it might sound, just because some of the layers aren't necessarily invertible in this sense. But we want to get there, and certainly these words don't explain all the variance.
I'm wondering if you mentioned first the where explanation.
Yeah.
So I'm wondering what does the where explanation look like on those images? So does it kind of is it very obvious from the where explanation, not obvious from the word explanation? How is it?
Yeah, good question. I guess we haven't looked at the where explanation specifically here, but it's a good point. Oh, sorry. The question was, have we looked at the where explanation, compare that to the what explanation? So we haven't specifically done it for these images. There's also, we should do that for sure. There's also a subtle distinction between what's called local explanation versus global. And so a local explanation says like, why is this image malignant? And that's where like a heat map might come in and saying, a global explanation is like, how do you do this task in general? And so that's what we're kind of doing here is like round lesions tend to be more benign. And so that also makes the comparison a little trickier, but I think it does point back to this kind of empirical evaluation of these explainability methods and see what is actually helpful to clinicians. All right, well, cool. Happy, I think I have my email up here. So happy to answer any more offline, and yeah, thanks.